Well, hello everyone. Welcome to Learn Wiko. Uh, Today we'll be discussing on digestion of nucleic acid, and we'll be covering a lot of scoops in this section. And before we start off, let's build on what you have known. So, what have you known about nucleic acid? Well, you must have known that nucleotides are the precursors of all nucleic acids, and also you must also have known that nucleotides contain a nitrogenous base, which might be a pyrimidine or a purine base. And also, you must have known that it also contains a pentose sugar, which is a 5 carbon sugar, and finally, a phosphate group, which is always esterified to the sugar. And also, we also know that nucleic acid also helps to store and transfer genetic informations, and the ability to help and store and transfer genetic information is evident in human DNA and RNAs, in which we have all known from our bases. But also, that is not all. Too, the purine and pyrimidine bases also have their own components too. The purines having the adenine and the guanine, and also the pyrimidines also containing the cytosine, the uracil, and the thiamine. And these components of the indigenous bases all have their own different functions and specificity. And this takes away from that. And that's not all. So, what you have also known, we must have known that the nucleic acids are also components of some coenzymes, which is NAD and FAD, nucleotinamide and flavinamide, are also some regulators like CAMP and CGAMP, and these are also what nucleic acid is composed of. Now, let's quote what we have today: digestion of nucleic acid. Now, check this: when we eat a food containing a molecule of nucleic acid, they are hydrolyzed to a mixture of nucleotides, and the key enzymes that plays a active role in these are the ribonucleics for ribonucleic acids and deoxyribonuclease for deoxyribonucleic acids. And this digestion starts in the stomach by the endogenous enzymes. And now away from that, um, also we take a step at the digestion of nucleic acid. The nucleoproteins will give us the nucleic acid and the proteins, and these proteins may be protamines or histone proteins. And this nucleic acid, given as a result of breeding of nucleoproteins, will give us nucleotides with the enzyme nuclease, as you can see in the chart. And that should give us the next reaction, which involves nucleotides given for the preceding reaction losing a phosphate group, sorry, by the action of the enzyme nucleotidase. When this happens, we are being left with our nucleoside. And don't forget, the nucleoside is a combination of the base and the pentose sugar. And now, what happens to the nucleoside after we have gotten it from the nucleotide? That's why we have the nucleosides, which will also be acted upon by the enzyme nucleosidase. When this happens, also, the nucleoside will be hydrolyzed to nitrogenous base and free pentose sugar. Well, when this happens, the nitrogenous base and the free pentose sugar are now free from their clustered components of nucleosides and nucleotide, or where we're learning along. And the nitrogenous bases then will then be catabolized. And when these are catabolized, this is a different pathway entirely, which we'll be studying in subsequent videos. Now, think about this now. If we have seen that we'll be having some free nitrogenous bases, now what happens to these free nitrogenous bases? I know you like to learn about this, I know. Now, this will take on to the next video. We'll be discussing more on that. So, up next, catabolism of nucleic acid. I hope you enjoy this. Keep subscribing. Thank you.